Hi everyone, welcome back. This unit's going to be about epidemiology mainly. So epidemiology is the study of infectious disease. This is a field that has developed considerably over the years. We've seen a lot of major changes in a relatively short period of time, mainly thanks to the development of cell theory and germ theory. So before we had our modern understanding of disease through germ theory, we believed that disease was caused by a variety of other things, primarily bad air. This is what the idea of miasma theory was at the time. All different forms of disease, infectious or otherwise, were ultimately believed to be caused by ailments in the air. That the air could be sick and you could catch disease by breathing it in or being exposed to it. The idea that disease could be caused by microorganisms, this was not a popular idea. And for a lot of time, we didn't even realize that microbes existed. Now, Louis Pasteur ultimately overturned a lot of these ideas. He refuted the idea of spontaneous generation and advanced germ theory, and germ theory would later go to replace miasma theory as the leading theory on disease. His rival, a German researcher named Robert Koch, created a framework for understanding and identifying the causative agent of these diseases. And this work, along with a lot of others, pushed forward germ theory as the current concept of infectious disease. And this became the foundation of epidemiology. Now, some important terms as we talk about infectious disease. We need to understand infectivity, pathogenicity, and virulence. Infectivity is the ability or likelihood of a pathogen to infect a host after exposure to that host. So if you are, say, walking around the street and somebody with a cold coughs or sneezes and the viral particles go into the air, and you get exposed to those viral particles, what is the likelihood of that pathogen managing to infect you? That is infectivity. Pathogenicity, on the other hand, is the ability of a pathogen to cause disease in those who are infected. It is possible to be infected with a pathogen and not develop disease. Not all infections will lead to disease. A lot of these infections can be successfully fought off without causing any actual illness. Virulence is a really important concept because this is the ability of a pathogen to cause severe disease, symptoms in those who are infected. This is a measure of the health impact that a pathogen has on the host. So the more virulent a pathogen is, the more damaging and costly to a person's health it is. Now, the study of epidemiology is pretty complex. There are lots of moving parts in the study of epidemiology, and we're going to dabble with and explore many of them. First and foremost, what happens when we are successfully infected and that infection turns into a disease? Well, we have a pretty common pattern of infection that we tend to observe. There are a few major stages of that disease. First, at the point of infection, there is a period of incubation. This is a period where we are not manifesting signs or symptoms of a disease, but the pathogen is reproducing in our body. Then we enter into the prodromal period, where we start to develop vague or general symptoms. We may start feeling a little bit not so great. Then we enter the proper illness phase, where we develop more severe signs and symptoms of that disease. At this point, we are reaching the apex of the number of pathogens that are present in our body while we are infected with that disease. Throughout this time, our immune system is engaged and is trying to fight this disease off. And it is the weaponization of our immune system that causes its decline, where we see a reduction in those symptoms and likewise a reduction in the number of pathogens. And eventually we reach the convalescence stage, where we finally go back to a period where we are no longer showing signs and symptoms and the pathogen is on its way out. Importantly, this is a variable time scale. This is just a general pattern that we observe, but the timeline that this can occur on can vary a lot. And since these are infectious diseases, there are periods of time during our infection that transmission can occur. As we live with and fight off this disease, the pathogens are replicating and they eventually need to escape your body if they're going to continue their life cycles. They need to go and find new hosts. And so there are periods of time where your body will be shedding these pathogens and you will be contagious. This period of time and when this occurs and how long it occurs for is also really variable from disease to disease. It can vary a lot based on the species that causes this disease and also the mode of transmission, how it gets around and reaches new hosts, and the mode of infection too. When it finds a new host, how does it infect that host? And more. It's important to remember that pathogens, as we talk about them, are just a form of parasite. These are organisms, or in the case of viruses, a non-living particle that sustains itself or perpetuates itself on the resources of a living organism. For this reason, viral particles are also considered parasitic. Even though they're not alive, they still exhibit this parasitic lifestyle where they steal nutrients and resources from organisms. They just use those things to build more of themselves. And we should ask ourselves, what is the goal of a pathogen? What are they doing and why? It's not just because they're truly evil beings. This is all just biology. So there is some underlying reason, a biological logic for why these organisms live this way. Pathogens are driven by this basic biological need to reproduce. It's something that all living things ultimately do. Even if we, for example, never have children in our lives, our cells still perpetuate themselves. All living things have this basic function. And their method to achieve this, however, just happens to result in the damage to the health of their host species. Their goal is to complete their life cycle and continue their lineage to make more of themselves and spread further to new hosts. This pattern has a number of key points that have to happen. The pathogen has to find a viable host organism. When it does, it has to be able to enter the host's body and find a home among the host's cells. It has to gain access to the body's nutrients. In the process, it has to also evade detection, elimination, or removal by the host's immune system. Then they have to reproduce a lot. They need to make many copies of themselves, as many as they can. And importantly, these copies have to be able to escape this host, the person or organism who is currently infected, and find a new host in order to restart this cycle and continue their lineage. To do this, pathogens have evolved many different traits to help them live this pathogenic lifestyle. For example, a lot of bacteria and viruses have special adhesion factors that allow them to grab a hold of and attach to a surface. These are called adhesins. Special chemicals that allow them to stick onto a surface, hold onto complementary receptors, special proteins that are on the host cell. Now remember, on the host, these special receptors that these bacteria are grabbing a hold of, they're not there for the bacteria to attach to. They serve some other function that is important to the host cell. The pathogen has just evolved to utilize them as a point of adhesion. If the pathogen's adhesin doesn't fit that host's receptors, there is no adhesion. There's no attachment, and likely that means the pathogen can't initiate infection. This is a factor that contributes to the specificity of a pathogen to a specific host. These bacteria and viruses steadily evolve to become better and more specific in their infection. They get more effective at parasitizing specific hosts as they get more specific about what they can attach to. In these situations, these adhesins will only bind to specific receptors, and so the bacteria that have them can only infect certain hosts. A good example of this would be Neisseria gonorrhoeae. This is the causative agent for gonorrhea. This is a species that utilizes some pretty specific adhesins that attach to receptors only found on the cells of the urethra, the vagina, and on sperm cells. Other body cells lack these receptors, and so a gonorrhea infection is pretty localized to the genitalia. To gain access to certain nutrients they need, pathogens have to use a variety of tactics. One tactic that a lot of bacteria and fungi use is to dissolve individual cells or attack body structures, break them down and digest their raw materials, or otherwise access their nutrients. To do this, a lot of these organisms can produce exotoxins. These are toxins that are secreted by the pathogen. They leave the pathogen's cell and are released into the environment to directly attack or destroy or otherwise break down host cells. There's a variety of types. You could have cytotoxins that will kill host cells or otherwise impact their functions. You could have neurotoxins that directly or indirectly interfere with the function of your neurons. You could have enterotoxins that damage or impact the cells that line your gastrointestinal tract. We're already familiar with one of these special exotoxins called hemolysins. These are specialized toxins that will attack and lyse host red blood cells. They'll break them apart into small pieces. This is the compound that does hemolysis, the same stuff that we see in our blood agar plates. And then we also have membrane-damaging toxins. These are special toxins that will interfere with or damage a cell's plasma membrane. A hemolysin is a type of membrane-damaging toxin. These toxins can result in the lysis of a cell. We can also see that some bacteria produce endotoxins. These are special compounds that are embedded on or in the pathogen's cell, and they're not secreted. A lipopolysaccharide, or an LPS, is a great example of this, a specific one that we find in gram-negative species. These endotoxins are released when the cells die or are digested, or sometimes when they undergo division. And when this happens, they get released into the environment. And so in particular, cells that attack them, like your white blood cells, can be affected by these endotoxins. Of course, avoiding the body's immune system is a really high priority. Pathogens that fail to avoid the immune system are attacked and disposed of. So many pathogens evolve strategies to deal with this, including avoiding being captured by your phagocytes, these special white blood cells that will move and hunt through the body and chase down and swallow cells that don't belong there. Other strategies involve avoiding being digested by the phagocytes. So if they get caught, they resist being broken down. Another strategy can involve avoiding antibodies binding to the cell. That's the main mechanism of antibody function. When the body produces antibodies, they target a specific feature on the foreign cell, and that's the thing that allows it to bind to. If the antibodies can't recognize those features, there is no binding. And so, antibodies are not effective on that particular strain. Some pathogens also evolve strategies to break down antibodies in the cell's environment. And there's a variety of other strategies that pathogens can evolve. This is just scratching the surface, really. Now, I want to discuss the spread of disease, and I'm going to talk about it with an unusual case. We're going to deviate a little bit from our typical discussion about human pathogens and talk about a plant pathogen, chestnut blight. Now, today, we don't see a lot of chestnut trees around. And there is a reason for that. Historically, Eastern North America used to be loaded with chestnut trees. It was a dominant feature of this landscape. The American chestnut was a widespread tree that occurred in a wide variety of habitats. There were estimated billions of trees across this range. However, this population shrank due to a few features. Logging was, of course, a major problem early on, and it certainly did not help their numbers, but it wasn't the greatest threat to the chestnut as it turned out. A fungal disease called chestnut blight was introduced to America. Now, this fungus is something that is endemic or native to East Asia, and it was introduced through imported Japanese chestnut trees. It was first introduced in New York City in the 1900s. It was brought to the New York Botanical Garden out there, and from there, the pathogen escaped to attack other chestnut trees in the environment. Now, this disease, it causes red patches and cankers to form on the trunk of the tree. And these patches can gradually grow and spread to other parts of the tree. And this disease can persist for months and months. It's got a very high mortality rate. So when a tree is infected, it is very likely to die of that disease. This instance of the disease being introduced or occurring in this new environment is what we would call an outbreak. It's the occurrence of a disease in a significantly greater abundance than what is expected in that region. It wasn't known from the region, and so it was introduced. Its expected occurrence is 0. So any instance in that case is an outbreak. Now, outbreaks can occur when you have a newly introduced emerging disease. Something that was not from this place, it has now been brought to a new place. It could be novel to a region. It could be novel to a population. So American chestnut trees have never seen chestnut blight, and suddenly they're being exposed to it. An outbreak could also be caused by a re-emerging disease, a disease that was already present in the region and already experienced by a population, but had been occurring at low rates and has now increased in occurrence. These outbreaks, an outbreak is an unusually large number of cases for a particular disease. And an outbreak can end when the frequency of that disease returns back to that expected level. If an outbreak continues and those infections start to spread to many, many, many other individuals, an outbreak can turn into an epidemic. An epidemic is the rapid spread of a disease in a population or a region, particularly if it occurs in a very short period of time. In an epidemic, we see the proportion of infected individuals grow rapidly. Zika and swine flu are good examples of epidemics in the recent past. This stands opposed to outbreaks that never really reached epidemic levels, like monkeypox, for example. When a disease persists for long enough in a population, it can become an endemic disease. Not epidemic, but endemic. Endemic in biology has two similar meanings. In biology generally, it just means that it's something that is native to a particular region. So American chestnuts, for example, are endemic to Eastern North America. In epidemiology, we can use this term in a different way. We can use it to say that a disease is constantly present in a particular region. It naturally occurs in a subset of that population now. So now chestnut blight is considered endemic to North American chestnut trees. Endemic diseases can still have outbreaks, importantly, and they can also still become epidemics. It's possible for that to still happen, even though the pathogen is considered now native to that area. Now, a pandemic is a disease that infects organisms across a huge range, across a massive geographic area at the same time. Typically, we consider something a pandemic when it occurs across multiple continents and is no longer bound by just one. The qualities of a pandemic are sometimes a bit debated or hard to describe. It's kind of one of those things that you know it when you see it. HIV, COVID-19, these are examples of pandemics that have occurred in the past. Now, pandemics are extremely difficult to combat once they happen. So if we can deal with a disease outbreak before it becomes an epidemic, and especially an epidemic before it becomes a pandemic, that can really increase the odds of success in preventing some pretty significant catastrophe.